Now let us move on from carbohydrates to focus on lipids. So a lipid is very similar to a carbohydrate in that it consists of carbons, hydrogens, and oxygens, just like a sugar does. However, in general, lipids do have a lower proportion of oxygen than carbohydrates. So this has a very interesting reaction. So it basically makes it very insoluble in water, and it also makes it a lot more dense. Okay, it means we can pack a lot more things into lipids than what we can carbohydrates. So some very important lipids that we know of are waxes, vitamins as well, and mono, di, or triglycerides. Now, lipids also have a wide variety of different roles inside of the body. So not only are they a brilliant energy source, but they also provide us with protection. They also are used in cell signaling. So they're used as a type of hormone. And they're also seen inside of our cell membranes. Now, lipids are constructed from what are called fatty acids. So a fatty acid is the simplest form of a lipid, and this can come as a triglyceride, which I'm going to explain in a moment, or a phospholipid, which is essentially just a lipid that contains a phosphate group. And this is what we see in our cell membrane. Now, something very, very important to note and have memorized is that a fatty acid, if it's unsaturated, so if you've got unsaturated fatty acids, it means that it contains a double bond somewhere throughout that fatty acid. If you have a saturated fatty acid, it means that it contains 0 double bonds, okay? It is completely filled up with single bonds only. So a good example of a unsaturated fatty acid is, I'm sure you guys have heard of your omega-3 and your omega-6. You see them in your fish oils. Those are unsaturated fatty acids. And that omega-3 or 6 tells us that it has a double bond at the 3rd or the 6th carbon. So now when you see an omega-3 or omega-6, you'll understand why it's called thus. So let us focus a little bit more now on triglycerides. So triglycerides are made primarily of 2 components. So we have the glycerol backbone, which I'll explain in a moment. And on that glycerol, we have 3 fatty acid chains connected to it. So this glycerol backbone is the same in all triglycerides. So this is our glycerol layer here. So you do not need to have memorized the structure of glycerol, nor do you need to memorize any of the structures of these biological molecules. It's very important for you instead to be able to differentiate between them and be able to understand what their role is, what they do inside of the human body. Now, you will see here that the reason why that we state that glycerol acts as like the backbone of a triglyceride is it allows these 3 fatty acids to attach itself to this glycerol backbone. So that way they are all grouped together as a triglyceride. Now, triglycerides provide more than twice as much energy per gram as carbohydrates and proteins in our body. So that's really, really important. This is why our bodies really like to eat fatty foods, because our body, through years of evolution, knows that our bodies rely on fats to provide a lot of energy in case of times where we don't eat much. So what it will do is it will try and build as many or get its hands on as many of these triglycerides as it can, because it has just so much energy per unit mass. Whereas carbohydrates, yes, it does provide a lot of energy, it is nowhere near as effective as triglycerides. Now, our bodies also have a virtually unlimited store of being able to store these triglycerides within our body. So, for example, I can't give you endless amount of sugars because your body can't store them all, it can't get rid of it, and that's where we result in type 2 diabetes. Whereas triglycerides, if you guys look at an adipose cell underneath a microscope, these are just big, plump cells that sole role is just to store these triglycerides. So we have more than enough capability to store and hold these triglycerides for a very long time. So now that we're finished with lipids, let us now move on to proteins. So something too that must be noted is proteins are massive. They are massive. They're absolutely huge. So if we have a water molecule, okay, water weighs 18 grams per mole. So don't worry, we haven't covered moles yet. But if we had 1 mole of water, it would weigh 18 grams. If we had 1 mole of the largest protein in the human body, it would weigh over 3 tons. Like, that's massive. You know, it's absolutely mind-boggling. So this just shows how huge proteins are inside of our human body. Now, proteins are also a lot more structurally complex than lipids or carbohydrates. Okay, they're a lot more confusing and they're a lot more diverse. But in saying that too, proteins also have a much more critical role inside of the human body. They are crucial, and that's a key word there. It is crucial to a wide variety of different cellular functions, whether it's structural, so it's building these structures to hold our body together, whether it regulates these cells and how they work. It's also seen in our immune system to protect ourselves from outside invaders. We see it everywhere. Now, the monomer of a protein, or the individual little subunit of a protein, is called an amino acid. Okay? Now, there are 20 different types of amino acid out there, so please do not worry memorizing it. For a lot of you, you will have to do that next year. So don't worry about memorizing all 20 of the amino acids and their structures and everything like that. That's not important. What we want you to do is just remember that amino acids are a monomer for proteins. So what that means is a good way to sort of visualize it, and this is how I visualize it, is picture your monomers as like Legos, as little Lego blocks. And we use those little Lego blocks, build them together in a certain way to build a certain structure. So we can get a heap of these little Lego blocks and build a toy car. Then we use that toy car, we get a bit bored of it, we don't need it, it doesn't work that well anymore. What we do is we break down that Lego car into its individual pieces, and then we build something else. And this is exactly how proteins work inside of the human body. So we get its monomer, so we get amino acids, and we join them together to form these big protein structures. Then, once that protein, we no longer need it anymore, it gets a bit old, a bit worn and torn, we can then pop it into our lysosomes, break it apart into its individual little amino acids again, and recycle it and reuse it to form different proteins for different functions. Now, something else that is very important for you guys to understand as well, like what we were talking about with carbohydrates, how they were joined together via a glycosidic bond. So that's using basically two hydroxyl groups to join together in a condensation reaction to join these 2 carbohydrate groups together. With proteins, it does it slightly differently, and that within an amino acid, we have 2 very different groups. So the first one is an amino-based group, and that is here. So amino is represented by NH2. And we also have a carboxyl or carboxylic acid side group. And a carboxylic acid is COOH. Now what happens is that proteins are created by joining these 2 amino acids one at a time. So how they join is, if we look at glycerine and alanine, what happens here is that we also have a condensation reaction. So we have a condensation reaction occurring, but it occurs between 2 amino acid groups. So we can see that we have our amine group on one side, and we have our carboxylic acid group on the other. And what happens is we have the hydrogen from the amine group bind with the hydroxyl group for the carboxylic acid, And what that will do is that will bind them together like thus. And it will combine them together. Now, this is called a peptide bond. So, like with carbohydrates, how it had a glycosidic bond, this here refers to a peptide bond, and that is what allows these proteins to hold themselves together. So, with a condensation reaction, what happens is that we can have these two amino acids joining with carboxylic acid on one end and the amino group on the other end. Now, it's very important too to note that the sequence of amino acids in a protein determines its shape and function. So, the way that these amino groups bind together is called the primary structure of a protein. Now, again, it's not important for you guys to know all four different sort of levels of structures for proteins. That will come in subsequent studies. However, it's very important to note that the reason why we have proteins in a particular order is so they will make a certain shape. Now, with almost all of chemistry, shape and the way it's all bound together relates to its function and its chemical properties. Now, let us look now at a very, very important type of protein, and this protein is called an enzyme. Now, it can be very tricky to try and adjust those two parameters to increase our chemical reactions inside the human body. So we can't just go, okay, I want to react now. I'm going to heat myself up to 90 degrees. Or some things need to be heated up to 500-600 degrees. We can't do that. We'll obviously die. We also can't control the concentration. We can't turn around and say, I need a lot of chloride ions in my body for this reaction to go a bit quicker, so I'm going to eat a heap of chloride. That's crazy poisonous. So that's another thing. So how do we increase our reaction speeds without basically cooking ourselves to death or poisoning ourselves? And the answer is with a catalyst. So, and this is very, very important. This must be memorized. Absolutely must be memorized. Is that a catalyst is a molecule that speeds up a chemical reaction by lowering the activation energy. Okay? That is really, really important. So how this works is that it helps lower the amount of energy needed for a reaction to occur and to take place. Now, common student trap that we see here is a lot of students think that a catalyst is involved in the reaction because it's speeding it up. No deal. Okay? So a catalyst will speed up a reaction but will not take part in that reaction. Now, a couple of things that are very important for you to know about enzymes. The first one is enzymes are crazy specific, okay? They target one thing and one thing only in a very specific and targeted way. Otherwise, if they weren't targeted, all hell would break loose. So it will only bind to a specific substrate, it will only bind to a specific molecule to induce its effect. The second one too is that enzymes are incredibly efficient. So for any of you who have opened up a bottle of red wine and sort of had a glass and not put the lid on properly, and you sort of come back to it after a week or two, maybe a little bit longer, and you notice that it tastes very bitter. It tastes very much like vinegar. And what happens is that red wine can sort of degrade over a long period of time to form vinegar. So this, which could normally take the space of several weeks, if not months, if we put an enzyme in there, it could happen in seconds, if not faster. So that's, I hope, puts it into contrast as to how quickly enzymes work. Now, another point too, and probably one of the most important, is that enzymes are subject to cellular control. Now, this is very, very important because it means that we can turn enzymes on or off depending on what we want to do. So if we want to use an enzyme to speed up the production of something, so we make it we put it in our little factories, it speeds up how quickly we can make what we want. And then suddenly we go, okay, brilliant, I've got how much I want, I don't need any more. So what we can do is, instead of having that enzyme that's still churning out a lot of that product, and we're going, whoa, stop, we don't want any more of this, we can turn those enzymes off so that they stop speeding up these reactions and they stop making more of these products. Okay, let us now move on to looking at nucleic acids. So nucleic acids are the largest molecule in our entire body. Okay, if we were to get all of your nucleic material inside of just one cell, and we connected them all together and stretched it right out, inside of one cell, inside of your nucleus, there is 2 meters worth of DNA. So that is how tightly that we squish and compact it in. And it also just shows that it is the largest molecule in the human body. If you guys have a look out for the Learning at Griffith page, the Griffith Health Student Success page, I'll actually upload a video on how you guys can extract the DNA or nucleic material out of strawberries with just common household ingredients. So nucleic material, we often see it in 2 different classes. So we see it as DNA, which is deoxyribonucleic acid, or we see it as RNA, which is ribonucleic acid. So DNA is the blueprint of life. It basically is responsible for coding and is the entire instruction handbook for making you. So picture DNA as like a library. So different sections of the library will code for different things, and this will tell your cells, which is the factory, how to build you. So how to make new skin, how to replicate, how to communicate with your body. Now, of course, DNA is very, very fragile. Okay, if we don't want to have any mistakes, any errors inside of our DNA, because that's where we have our genetic deformities, and that's a big no-no. We don't want that. So instead of constantly prying open and reading through our DNA to get our make a copy to turn it into proteins, instead we use what is called RNA. So RNA, think of it as a temporary copy of DNA. So this entire process, which I'll explain again in a moment, is called the central dogma of genetics. So when we want to create something, whether it's more proteins, so for example, let's look at hemoglobin. So hemoglobin is a key protein that is seen inside of red blood cells, and it's hemoglobin that actually holds on to the oxygen and transports it around the body inside of red blood cells. So let us say that we want to make some more hemoglobin. So what we do is we initiate essentially transcription. Now, don't worry about that process just now, but transcription is the process of converting DNA to RNA. So picture it, if you will, that we go into our library, we pull out the book on how to make hemoglobin 101. Now, we don't want to just sort of check this book out and leave, because that book is valuable. We only have the one, and if we break it, that library, which is your cell, can no longer create hemoglobin properly. So what we do is, instead, we get our book and we make a photocopy of it. We make a temporary copy. Now, of course, we're not going to take a photocopy and stick it up on the shelves in a library, okay? But we can then use that photocopy as an instruction guide on how to build hemoglobin, and then just chuck it away when we're done with it. So this way, by copying the DNA to RNA, or to get our book and make a photocopy of it, it means that we get a temporary copy of our instructions without any risk of breaking or making any errors. So what we do is, once we have our RNA, or our temporary photocopy, we can then send that temporary copy to the ribosomes. And what that does is it gives that copy to the ribosomes and says, okay, here is the sequence of amino acids. Here's what we want. And what it does is that these ribosomes will then get our amino acids, like we discussed earlier, and join them together in the correct order. Okay, so once we join them all together, we have the correct sequence, it can then fold, poke, and prod it, and there we have our hemoglobin. So, nucleic material is very, very important inside of our bodies, as without it, we can't exist. So every single living thing, whether it is the biggest of elephants to the smallest of viruses, all contain nucleic acid. For without it, life cannot exist. This is why plastic isn't alive. This is why my pen isn't alive, because it doesn't have any nucleic material. Now, this debate on whether viruses are alive or dead is a very complicated one, so I don't want you to get sort of too intertwined into that. I just want you to be aware that every single living thing does contain nucleic material, and it is paramount to being able to survive and procreate. Now, with our nucleic material, the structural unit, so the things that make up our DNA or RNA, is called a nucleotide. So, for example, with carbohydrate, how we had our saccharides, for lipids, how we had our triglycerides, and for our proteins, how we had amino acids. For nucleic acid, we have what are called nucleotides. Now, these nucleotides are comprised of 4 different compounds. And these are called adenine, thymine, cytosine, and guanine. Okay? So, and this is where, for me personally, this just blows my mind. So, all of DNA, the trillions and trillions of billions and zillions, squatillions of base pairs within not only myself, but all diversity of life, whether it is a bacteria, whether it's a human, boy, girl, whether it's the difference between a giraffe and a pineapple. It all comes down to the different combination of these 4 different compounds. And that, I just find that absolutely amazing, in that depending on how these 2 groups join together, because A can only bind to T and C can only bind to G. So it's just the order and combination of these 2 forming our genome that creates the diversity of all of life as we know it. And I hope you find that as equally as amazing as I do. Okay, guys, so this now wraps up Module 6, Video 1. Thank you all very much for watching. I hope you enjoyed it. If you do have any questions or queries regarding this video, please feel free to send me an email at b.grant@griffith.edu.au. If I could also get you to please do the survey down the doobly-doo, the little the description bar down below. There is a little link if you can just click on that. It's a very short sort of 4-question survey. And what that does is it tells me a lot about what you think about these videos, whether you enjoy it or not. And also gives me a lot of feedback on how I can constantly improve and make these videos better to sort of assist in your studies. So thank you all very much for watching. My name is Brock Grant, and I'll see you all next time.